Hello, can you hear me? Thank you.
ম্যাডাম ভিডিওটা অন হচ্ছে না, ভিডিও অন করতে পারছি না। ওকে ম্যাডাম, ওকে ম্যাডাম।
Thank you, Dr. Papir Ghosh. I am taking this privilege to introduce Dr. Budhaditya Mukherjee from IIT Kharagpur. Dr. Mukherjee, you please on your camera so that everyone can see you.
Yeah.
Okay, but I am not actually. I don't know. It's okay. Well, good afternoon. It's my privilege to introduce Dr. Budhaditya Mukherjee from IIT Kharagpur. Dr. Mukherjee, who is presently the Assistant Professor of School of Medicine, Medical Science and Technology, IIT Kharagpur. Dr. Mukherjee had his master degree in the genetics from University of Calcutta. Then he completed his PhD from Department of Infectious Disease and Immunology, Indian Institute of Chemical Biology, that is ISAB Kolkata. Then he joined as a postdoctoral fellow to Department of Microbiology and Molecular Medicine, University of Geneva, and also from Department of Microbiology and Immunology, Johns Hopkins Bloomberg School of Public Health. Dr. Mukherjee have excellent academic record. He has received many awards like Pfizer Research Award in the area of infectious disease, rheumatology and immunology, INSA Medal for Young Scientists, Swiss Government Excellence Scholarship, EMBO Long-Term European Fellowship, Parafrap International Fellowship, CSIR Indian National Eligibility Test Fellowship. He has industrial collaborations with UCB Pharma SA and Medicines for Malaria, funding secured 4,000 USD. Also, he has many seminal publications in the field of molecular parasitology and immunology in the very reputed and internationally acclaimed journal like EMBO Journal, Science, eLife, Journal of Immunology, and PNAS, PLOS Neglected Tropical Medicine, EMBO Molecular Medicine, and International Journal of Parasitology. With these few words as introductory note, may I request Dr. Mukherjee to start his deliberations to our students who are eagerly waiting for words from your end. Thank you.
Yes, sure. Yes, hello everyone. My screen is full, right? You guys can see the full screen. Okay. So, firstly, thank you so much for having me. Yes, it's a very nice thing to talk with the students. And since this is particularly a student orientation program, so I have designed my presentation in such a way that I will give a light, although it's already a very nice introduction from Dr. Ghosh for my introduction and my publication, but I thought that I will be more elusive in the manner and I will show them the career path that I took just to give them an idea. So today I am going to talk about the fundamental role that played by a particular enzyme in Apicomplexan parasite, which enzyme is called aspartyl protease, that plays an important role in the establishment of infection by these parasites. But before I go into that main topic, let me just give you a brief idea about my research career. So after my MSc in genetics, I started my PhD in CSIR ISAB. And this point, I was working on infectious disease. And at this point, I didn't know. But after that, my entire career have been circulating among different aspects of these infectious diseases. So during my PhD, I was working on host-parasite interaction with respect to Leishmania parasite. You know probably that this is a kinetoplastic parasite and it causes visceral leishmaniasis. So I was working on a particular species, which is called Leishmania donovani, and this is fatal if left untreated. But I was more focused on the drug resistance problem, which I will talk shortly during this presentation also. So after I did my PhD, I went for a postdoc in US in Johns Hopkins, where I was there for a short period of time. And at that point, I was working on something very similar to what I was doing in my PhD. I was working on investigating the role of a host cytokine molecule. Basically, these are the regulatory molecule that hosts produce in response to any pathogen or any kind of immune response from the side of the host. And I was working on the role of the IL-10 in the colitis model. After that, I joined University of Geneva, Dr. Dominic Soldati's group as a postdoctoral fellow, and this was in 2015. And I started working on Apicomplexan parasites. This time I was working more on the cell biology and biochemical aspect of this parasite, and mostly the work I present today will be on this part. And after that, I came back to India in 2019 and joined IIT Kharagpur as an assistant professor in last year June, and till then I have been here. So before I talk about any kind of host immune response, let me just explain this to you. So in response to any pathogen, when I'm saying pathogen, I'm mostly referring to intracellular protozoan pathogen, which cause human infection, for example, trypanosome or leishmania. Now, when they infect the host cell, they are usually taken up by the antigen presenting cell, which could be a macrophage or dendritic cell. And the unique property of this cell is that they can process these parasites into small, or the protein from this parasite into small fragment, which are then represented on the surface of these cells via MHC2, typically, unless it's a viral infection. And then after this antigen is represented on the host surface, it results in the activation of the adaptive immunity, which mostly consists of the effector T cells, CD4 positive or CD8 positive T cells, which then produce these cytokine molecules to regulate the infection, or a cytotoxic killer T cell to basically kill the infected cells. So this intracellular pathogen have developed many ways by which they can bypass this host immune response. So you are probably aware that, I will talk about this first. So you are probably aware that several pathogens, for instance, in this case, if you see that I have represented several pathogens like trypanosoma, leishmania, toxoplasma, what is common feature among these pathogens they have all evolved a way by which they can grow and replicate within the host cell. and they can basically evade the host immune response or any immunity challenge from the host. So, for example, trypomastigotes, which is basically the promastigote form of the trypanosoma, when they enter the macrophage or any antigen-presenting cell, they recruit the lysosome on the surface of the host membrane, which allow this parasite to enter the host. If you see about Toxoplasma, these have got several secretory molecules which it secrete as it enters into the host, and some of these molecules can go into the host nucleus, thereby modulate the host function to create a perfect niche where this parasite can grow and replicate. I will roughly talk about one of these strategy employed by this protozoan parasite, Leishmania. So what this parasite do is, it when it goes into the cell, it destroy or affect several antigen processing machinery as well as antigen presentation machinery, so that its antigen are not represented on the surface of the host cell, and hence there is no adaptive response or the T cell response, as I have shown you previously. So if you being aware of this life cycle of this parasite, you must be know that this is a diagenetic parasite having two hosts, basically a human host and a sandfly host. So normally this sandfly inject the promastigote form, which is the motile form for this parasite, to into the human skin. And these parasites then infect the human. They mainly affect the liver and spleen of this human. And then this infection spread, and during an infected bird blood meal, the sandfly can again capture the amastigote form from the blood of this infected host. And within the sandfly, they get converted into promastigote. So you can see clearly when these promastigotes enter the host, they have a need to evade host immune response, as well as now there is an emerging concept coming out, that is when they goes into the sandfly vector, or for other pathogens the mosquito vector, there is also immune response challenge in that vector. So these parasites are successful because they can successfully evade this immune response, and they have developed a way to fool basically host immune response so that they can survive within the host. Now, my PhD work was particularly on the Leishmania parasite, but on the drug-resistant form of the Leishmania parasites. Now, if you look at this map, it's a map of the hyper-endemic zone of Bihar, and you can see that Leishmania, for the treatment of the Leishmania, antimonials were the primary drug that was introduced in Indian subcontinent in long back, 1921. And with its introduction, the drug was a dramatic success. So, but however, with the as the time progressed, as happens with many parasitic disease, the drug started to lose its efficacy. This is because of the emergence of the resistance to the antimonial drug, and with the emergence of the resistance, a new drug like metformin and amphotericin B came into the picture, but resistance against those drugs are also emerging at a rapid rate. So it was a very relevant problem to talk about that time, or to address particularly. And if you look at this picture, you will see that we collected these parasites all from this hyperendemic zone of the Bihar, and you can see that most of them, about 78% represented here in red, were showing unresponsiveness to the antimonial drugs. Not only that, if you look into the biochemical and biophysical properties of this parasite, you will see that apart from their responsiveness to the antimonial drug, they have certain biochemical properties which are quietly distinctly vary from their sensitive counterparts. For example, they have a different membrane fluidity, they express different sugar on their surface, they can modulate the host cell differently, and this is not a surprise. Actually, a parasite which differs so much in their biochemical and biophysical property can modulate their host differentially, thus resulting in an entire different disease outcome. So the problem in this respect we try to address is how antimony-resistant Leishmania donovani results in an aggressive pathology and results that ultimately affect its disease outcome. And to address this, so we selected the parasite from the hyperendemic zone of Bihar to compare them on their drug resistivity. And the major observation that we look into is this. If you look into the literature that was already available, you can see that when this antimony resistant parasite infect their host, they always results in a high number of parasite burden, meaning there is always a larger number of parasite loading into the liver and the spleen of the patient, and this is correlated with the high level of cytokine production in the host. And basically we try to address that issue. What we find out is that this antimony resistant parasite express a unique sugar on their surface that allow them to interact with host innate immune receptors, which in this case is represented as TLR2 and TLR6. So basically parasite, antimony resistant parasite, which express the sugar on their surface, allow them to interact specifically with this host TLR molecules that results in the activation of a signaling pathway, which ultimately results in the translocation of a transcription factor NF-kappa B into the host nucleus. As you can see here that the two subunit of NF-kappa B is represented in green and red, and as with the infection they goes into the host nucleus, you can see this color of cyan appearing for the merge of these two color. Now, as soon as they goes into the host nucleus, they doesn't bind with the IL-10 promoter immediately. Rather than they bind to a promoter which is represent promoter of a gene which is represented as microRNA. You know, probably you were aware that microRNA are small RNA and they basically bind to the 3' UTR of a mRNA, resulting in its degradation. Basically, they are responsible for normally, they are responsible for the downregulation of a particular expression of a particular gene. So what happens is this NF-kappa B transcription factor that binds within the promoter of this microRNA and thus results in the induction of this microRNA in the host cell. This microRNA goes and binds with the 3' UTR of the host MY88, which is a downstream molecule of the TLR, and this is a host, another host molecule, and which results in subsequent degradation of this MY88 molecule. As soon as this level of MY88 decrease, there is an increased IL-12 level in the host, and this results in a removal of the inhibition of the host IL-10 promoter, and the NF-kappa B transcription factor, which have been translocated into the host nucleus, now can bind specifically on a specific site of the host IL-10 promoter, thereby resulting in a huge amount of IL-10 induction. And what does this IL-10 do? This IL-10 in turn upregulate the expression of a transporter on the host surface membrane, on the host membrane, and this upregulation of MDR1 is responsible for resulting of efflux of antimony from the host cell. So you see that this resistant parasite have devised a unique way or designed a unique way by which they can get rid of antimony from the host, and thus they provide a selective survival advantage over the antimony sensitive parasite in the same microenvironment. Thus, it explains why we always see a high parasite load in the patients with antimony resistant infection as compared to the sensitive counterpart. Interestingly, as we were doing this work, another group was working and on the similar type of problem, and they found that the antimony sensitive parasite, which does not express this sugar on their surface, results in the deactivation of the host transcription factor, which include ERK and NF-kappa B. So this clearly suggests that these two parasites, on the basis of their biochemical property, have found a way to differentially modulate their host, thus resulting in a different disease outcome. As I have talked to you, the most important clinical significance of this observation is that this antimony resistant parasite, whenever it infects the host, it always results in a huge amount of organ parasite load and high level of IL-10 in the serum. And with this observation, we can link why there is a high level of IL-10 whenever there is antimony resistant parasite infection, and why does it provide a selective survival advantage to this parasite. Now, if we look at this clinical point of view, so what we saw that infection with antimony resistant parasite, which is represented here as SBR, or if you treat the cells, host cells with recombinant IL-10, it results in the upregulation of a host transporter on the surface of the host cell membrane, infected host cell membrane, as I have talked to you previously. As you can see here, whenever you give infection with antimony resistant parasite or treat with a certain dose of recombinant IL-10, you can see that this MDR1 expression on the surface of the host is increased exponentially. And what we could show that if we specifically target this SBR LD driven, antimony resistant LD driven IL-10 induction in the host by using small molecule inhibitors that specifically target this pathway of ERK and FKB dependent IL-10 induction in the antimony-resistant infected host, we can make this resistant parasite susceptible to the antimonial drugs once more. So that is what some of my major findings during my PhD. And after completion of my PhD, I moved into Geneva and started working on the Apicomplexan parasite, more on the cell biology and the biochemical aspect. Now, what are Apicomplexan parasite? Probably you were well aware that this is the largest group of intracellular protozoan pathogen, causing maximum number of death each year in the world after the bacterial infection. And if you look at this group, you will see that this Apicomplexan parasite not only consists of only human pathogen, but they also cause infection to large number of economically important animal pathogens also. But I have represented only 3 of them here. Plasmodium, which is responsible for causing malaria and 500,000 deaths each year. Toxoplasma, a parasite that have found a way to infect almost all nucleated cells, and a parasite which affects about 1/3 of the human population worldwide. Cryptosporidium, which in recent years has come up as one of the major cause of the child death in India after rotavirus. So basically, I started working on, excuse me. So basically, I started working on these parasites, particularly on the invasive stages or the infection stages of this parasite. Now, if we look at the invasive stages of this parasite, be it a Plasmodium merozoite or a Toxoplasma tachyzoite or a Cryptosporidium sporozoite, we will see that certain specialized secretory organelle like micronym and rhabdoid are present in all of them. Now, as in case of these parasites, as in case of any other Apicomplexan parasite, the process of invasion or the host invasion is a tightly regulated process which requires synchronized secretion from this secretory organelle, micronym and rhabdoid, that contains several proteins like adherins, perforins, proteases that allow this parasite to infect their host and complete their life cycle. What happens is the micronym protein are released first. So this allows this parasite to break the intracellular vacuole or the parasitophorous vacuole within which it is confined within the host. And then it also lies the host cell membrane and allows this parasite to escape from their existing host. Once they are out of this host, which we commonly refer to as egress, they start the process of reinvasion, a new host. And what happens during this time, the rhabdoid proteins are secreted. So this can be again divided into 2 parts, rhabdoid neck protein and rhabdoid bulb protein. So rhabdoid neck protein are released first, which results in the formation of a tight junction between the host and the parasite. And the rhabdoid bulb protein are then injected into the host, and this can go into the host nucleus or host cytoplasm, thereby modulating the host microenvironment and creating a perfect environment where this parasite can grow and replicate. Now, if we look at this process with the help of a video microscopy, you will see that this parasite glides as it come in contact with the host. It started the process of entering the host, and as it do so, it formed a bulb or moving junction between the intersection of the host and the parasite. And finally, it completes its process of invading the host. Now, if you tag a rhabdoid neck protein during this process of invasion, you will observe something like this, where you can see this protein is moving from into the rhabdoid, into the moving junction, basically, which signifies the role or the importance of this protein in the lytic cycle or the process of invasion of these parasites. Now, it is also important to remember that like any other protein, this rhabdoid and the micronym protein are synthesized in the secretory pathway, and they travel through the secretory pathway before they reach their final destination in the rhabdoid and the micronym. Now, what happens in this secretory pathway is that this micronym and rhabdoid protein are processed from its pro to mature form. And this processing is not only important for proper functioning of this protein, but also proper targeting of this protein to their respective compartment in micronym and rhabdoid. We could localize an aspartyl protease that is specifically present in this secretory compartment, where this processing of micronym and rhabdoid protein takes place. As you can see here, this aspartyl protease represented here in red. It was tagged with a tie, and if you see here with the green, which is a Golgi resistant marker, you can see that it is present just above the Golgi, thus showing its location in the ELC or secretory compartment where this processing of microneme and droplet protein take place. Now, what is aspartic proteases? So, aspartic proteases are endopeptides. They are present in all eukaryotes, including human. They usually have a pro region and an active site. Now, within their active site, they usually have a DTG motif. In some cases, this might be a DSG motif. So DTG represent amino acid here. And within this active site, there is a flap and flexible loop, which is mainly responsible for the ligand binding. So they have a broad range of function from enzyme maturation to signal transduction, or they can act as a virulence factor. And it is worthy in mentioning that these aspartyl proteases have always been a good choice for chemotherapeutic targets, particularly after the success story of HIV-1 aspartic protease inhibitors. So one of the major medicine to treat HIV is with aspartic protease inhibitor, against one of its protease, HIV-1. Now, if we look at the broad repertoire of aspartyl proteases in the Apicomplexan parasite, you will see that based on their function, they can be clustered into several groups. For example, you must be knowing about food vacuole or hemoglobin degrading plasmopsins like plasmopsin 1, plasmopsin 2, plasmopsin 4, and you can see that they can be clustered in one group. Similarly, ASP3 or TG Toxoplasma gondii aspartyl protease 3, which was localized in the secretory pathway of the parasite, also have two orthologs in Plasmodium, which we refer to as plasmopsin 9 and plasmopsin 10. So we wanted to study the function of these proteases. So we follow a classical genetic approach where reverse genetic approach where you try to knock out the function, or where you try to look into the function of a particular gene by creating a knockout. However, repeated attempt from our side to generate a clean knockout of these proteases were not successful. This we study with a simple assay, as I will explain here, which is called a plaque assay. Now, as I have mentioned to you before, that this parasite, when they invade into the host, they grow and replicate within their host for a short period of time, or a certain period of time, I should say. And once this replication and growth phase is over, they lyse their existing host, and as they do so, they results in the formation of these small white plaques, which are visible to us if you stain with crystal violet. Now, if you have defect in any one of the stage of this lytic cycle, you will not see the formation of these white plaques anymore. As you can see here, in case of wild-type parasite, you see that they lyse their host cell to form this white plaques. However, if you knock out the protease, you don't see the formation of these plaques anymore. This prompted us to go for an inducible knockdown system to study the function of this aspartyl protease. And what we did is, with the help of CRISPR-Cas9, please let me know if you have any doubt regarding this method, I will explain. But what we did essentially is that we replaced the endogenous promoter, or the original promoter of this aspartyl protease, with a Tate inducible promoter. So what happens is, normally this promoter is active and the mRNA is transcribed normally, and subsequently protein is produced. However, if you add ATC or anhydrous tetracycline, then this promoter is not active anymore. And as you can see here, in case of when you don't add ATC, or in case of minus ATC, which basically represent the wild type scenario, you see that this protein ASP3 is produced in its pro and mature form, represented here around 70 and 55 kilodalton. However, if you treat this with ATC, you see that this protein is not protein disappearing in the immunoblot. This is also evident from the immunofluorescence. As you can see here, in case of the wild type scenario, you see these red dots which represent the ASP3. However, if you treat these parasites with aspartyl, if you treat this parasite with anhydrous tetracycline, you see that this protein is gone. So once we establish this knockdown condition, the first thing we check is what is the result of this knockdown on the processing of the microneme and droplet protein, particularly because of the fact that this aspartyl protease is present in the secretory pathway where this processing of microneme and droplet protein takes place. Now, if we look here, in case of the wild type scenario, mixes represent is just one representative of a micronym protein. You see in the wild type scenario, this micronym protein mix6 is processed from its pro to mature form. However, if you knock down the protease, you see starting an accumulation of unprocessed micronym protein. Similarly, for the ROP2 protein, ROP1, you see that in the wild type scenario, you see that this protein is processed from its pro to mature form. Again, if you knock down the protease, you started seeing an accumulation of unprocessed ROP3 protein. And I must mention here that this mix6 and ROP1 are just two representative of micronym and ROP3 protein. This is true for the most of the proteins that we check. So the first thing we investigate is what is the effect of the accumulation of this unprocessed ROP3 protein on the lytic cycle of the parasite. This we did with the help of a simple assay, as we'd like to call it as an evacual assay. Now, as I have mentioned to you, these parasites, when they enter the host, they secrete their ROP3 content and micronym content within the host, which allows this parasite to basically invade the host. However, if you treat this parasite with cytochalasin D, which is an actin myosin inhibitor, this parasite will still attach itself on the surface of the host and discharge its ROP3 content within the host. However, as it is treated with an actin myosin motor inhibitor, basically it will fail to move inside the host. Okay, and as a result of that, you can see that this green trail of ROP3, which is discharged in case of the wild type parasite. However, if you knock down this protease, you don't see this discharge of the ROP3 content anymore. As you can see here, this green content or the ROP3 protein is mostly within the parasite and they are not discharged within the host. So what we understand is that as a result of the accumulation of unprocessed ROP3 protein due to the knockdown of this aspartyl protease, there is a defective ROP3 discharge and thus resulting in a defective process of invasion in this parasite. So obviously the next thing that we wanted to look into what is the result of the accumulation of the unprocessed micronym proteins, as you can see here, was having on the lytic cycle of this parasite. Now this I will try to represent with a simple video. Now if you look at the Plasmodium parasite, when they come out from the infected RBC, you can see that they can rapidly lyse the infected RBC membrane and these parasites are coming out from their host. This is slow down for you, but it's actually very fast and rapid process. Now what happens is when you knock down the parasite, and this is the Toxoplasma parasite represented in the infected vacuole, as you can see here. If you knock down this protease, you will see that these parasites are trapped in a joint ball or vacuole-like structure. They try to break this vacuole and come out of it. Eventually, one or two can do so, but most of them remain trapped in this ball or vacuole-like structure. As you can also see here, they are mostly remaining trapped in this big vacuole-like structure and fail to break the vacuole and come out of the host. So if I sum up this, so accumulation of the unprocessed micronym protein results in a defective egress of this parasite, meaning that they are trapped within their host. They cannot come out and reinvade a new host. As a result, normally the transmission of this parasite from one host to another is blocked. Now, to sum up, if I sum up this observation so far, I can say that AS3 acts as a maturase for the ROP3 and micronym protein that help in the process of invasion and egress in the host. And this work of AS3 is completed by two of its ortholog in Plasmodium, Plasmipsin 9, which primarily helps in the process of invasion, and Plasmipsin 10, which help in the process of egress. Now, as I have mentioned to you before, that this aspartyl protease always have been a preferred choice for chemotherapy. So people have been working to design small molecule inhibitors that can specifically target these aspartyl proteases. And the first aspartyl proteases that were characterized was food vacuole plasmipsin or hemoglobin degrading plasmipsins. So a large number of inhibitor molecules have been designed around the world to try as a malarial drug by targeting this aspartyl protease. As we are working on this project, there was a Swiss pharma company, Actilum, which at that point was an independent company and now a part of Pfizer, was working on a peptidomimetic inhibitor or a library of inhibitor that it have been designed against one of the food vacuole plasmipsins, plasmipsin 2. Now, Plasmodium 2, if you look in the expression profile of Plasmodium 2, it is normally expressed in the host at around 24-hour post-infection. And you can see that when they tested these two compounds among their library, 49B and 49C, with a fairly similar structure, both of them have a very high IC50 of 500 nanomolar around 24-hour post-infection, which clearly suggests that these compounds were not targeting Plasmodium 2. Hence, the company got not interested them in anymore, and they dropped these two compounds from their screen. But when we look into their data, we find something very interesting. One of these compounds, particularly 49C, have a very high IC50 72-hour post-infection. Roughly when the late stage plasmodium 9 and 10 were expressed. And this have an IC50 of 0.6 nanomolar. To mention to you that this is lower than artemisinin, which is currently used for the treatment of the malaria. Hence, we obviously got interested in this compound and decided to characterize this further. Now, if I look into the broad range of aspartyl protease that is expressed across the erythrocytic blood stage of Plasmodium parasite, you will see that food vacuole plasmopsins like plasmopsin 1, 2, 4, or HAP is expressed from the early ring to the trophozoite stage, which is basically the feeding stage for this parasite. Plasmopsin 5, which is another aspartyl protease that is mainly responsible for cleaving of the protein, which are then sent into the host nucleus, is roughly also expressed during that point. And plasmopsin 9 and 10, which are mainly responsible for the invasion and the egress of this parasite, are expressed in the late schizon stage. So what we did is we apply the compound early, either on the early ring or on the early schizon stage, and then allow this parasite to grow and complete their erythrocytic life stage, and recollect this parasite when they are able to break this vacuole and reinvade a new host, which is characterized by the formation of this immature ring. What we observed was very interesting. In presence of 1 nanomolar of 49C, which is a very low concentration, you see that these parasites are trapped in a joint, ball, or vacuole-like structure. Doesn't matter whether you have applied the drug on the ring stage or the schizon stage, they remain blocked in this vacuole-like structure, meaning like the DMS control where you don't treat them with drug, when these parasites are able to break this vacuole and reinvade a new host, which is characterized by the formation of this immature ring, you don't see that happen in the presence of the 49C. Interestingly, if you break this vacuole mechanically with the help of a syringe and allow this parasite to reinvade the host, they still fail to do so, suggesting that this compound not only block the egress of the parasite, but also block the invasion of the parasite. This clearly suggests that this compound is targeting late stage plasmopsin 9 and 10, which it helps in the process of ingress and invasion. Now, you can also conclude another more thing from this experiment. Now, the drug applied to the early ring, in case of the early ring stage of the parasite, does not stop it from developing into a mature schizon. So the parasite can grow and develop into a mature schizon stage. It is until when it reaches this stage, it gets blocked. So you can clearly imagine that this drug is not targeting any other food vacuole plasmopsins like plasmopsin 2, 4, and HAP, which talks about its specificity. Now, I will try to convince you also visually. So what we did is to see the effect of the compound, we tagged one of the parasitophorus vacuole protein, PP1, with a GFP. Now, what happens is when the parasite grow and accumulate within the parasitophorus vacuole, you see that the huge accumulation of the GFP or the green signal within the host. Now, as they lyse their host cell membrane and get out of the host, you see that this green color disappearing, which is basically due to the release of the GFP. Now, if you treat with 49C in 1 nanomolar concentration, you see that at time 0, this GFP is accumulating within the infected host. Now, after some time, you see that the GFP is disappearing. However, the parasite is still maintaining its circular configuration and intact vacuolar position. So basically what it suggests at this low nanomolar concentration, the parasite were able to rupture the parasitophorus vacuole membrane, but it still failed to rupture the host plasma membrane to get out of the host. As you can already understand that this is an intracellular pathogen, and for many intracellular pathogen, like many intracellular pathogen, it resides within the parasitophorous vacuole. So in order to get out of the host, it need to lyse both this parasitophorous vacuole membrane and the host plasma membrane. However, if you apply the drug at a high concentration, say 10 nanomolar concentration of 49C, you see that the GFP is lasting throughout this process, which clearly suggests that even at the high concentration of the 49C, this parasite even failed to lyse the host plasma parasitophorous vacuole membrane. So you can see here clearly that the two membrane and this parasite are failing to rupture the parasitophorous vacuole membrane. So we finally test the effect of this compound in mice. So for the mice, we use a strain of Plasmodium burgeri, which can infect the mice to basically study the effect of the compound. So in case of the wild type scenario, what you see is that when you infect these mice with the Plasmodium sporozoite, and these parasites were expressing luciferase, which is basically a signal that you can detect with the help of a luminometer, you can see that this luciferase signal is accumulating in the liver of this parasite, as you can see here in the blue, 44-hour post-infection. Now, if you follow a time course, you will see that this signal persisting in 52-hour post-infection. After that time, around 55 hours, there is a roughly disappearance of these parasites. This is the time when these parasites are leaving the hepatic cells in the liver and they're going into the red blood cells or infecting the red blood cells. Then the signal started to appear as the blood infection started to occur, and gradually the infection spread. Now, if you treat with 49C 20 hours post-infection and follow the similar kinetics, you will see that in presence of 49C, compound 49C, you still see the accumulation of parasites in the liver around 44 hours. Around 54 hours, you actually see a more intense signal than the control mice, which probably is because of the fact that these parasites are failing to go out of the liver cell, or they're failing to rupture the hepatic cells and they cannot go into the blood stage. So basically you see the signal is lasting even in 55 hours post-infection. Finally, these parasites started to infect or try to infect the blood cells. However, they have a massive defect there and the infection is cleared. So this clearly proved that the 49C is affecting the invasion and the egress of the parasite in the actual infection model or in the in vivo scenario. So if I sum this up, so we have a compound 49C, which is slightly different than a similar compound 49B. However, 49C can specifically target two plasmapsins in the Plasmodium parasite, plasmapsin 9 and 10, thus resulting in a block in egress and invasion of this parasite. However, its closely similar structure compound 49B failed to do so, which shows that this compound have a high effectivity. The question is that whether this compound can also target its ortholog of the plasmapsin 9 and 10 aspartyl protease in case of Toxoplasma. We wanted to test this to see whether this compound is also effective across the phylum. And we did that with the similar assays as I have mentioned to you. So we basically checked the processing of the micronym and the Rop3 protein, and as we have done previously, and you see here the RON4, which is a Rop3 protein. In presence of the DMSO, this protein is processed, like in the wild type scenario, from its pro to mature form. As soon as you started applying 49C compound, you started seeing that this protein is not being processed anymore, and you see that this the pro form accumulating more and more for this protein. This is also true for the MIG protein and also for the raw protein. As you can see here, in both the cases, the mature form are disappearing as you are increasing the concentration of the 49C. So in a simple way, we can clearly see that the 49C also results in the accumulation of the unprocessed micronym and Rop3 protein. Thus, it recapitulates the ASP3 knockdown phenotype. Basically, it targets the ASP3 protein in a simple way to say. Thus, we can see that this compound is targeting both aspartyl protease 3 in Toxoplasma and plasmapsin 9 and 10 in the Plasmodium. Now, we have already got the evidence of the specificity of this compound in Plasmodium. However, we wanted to further check the specificity of the compound in Toxoplasma. Now, as I have mentioned, plasmopsin 5, As5 is another ortholog of plasmopsin 5 in Toxoplasma. And importantly, like As3, As5 is also a Golgi resident aspartate protease. So they have roughly similar position in the parasite, and mostly As5 is responsible for processing proteins with a specific sequence. These proteins are then allowed to be translocated into the host nucleus, and GRAS is one of the major characterized substrate of this aspartate protease As5. So when the GRAS6 get processed by As5, it then goes into the host nucleus. So we wanted to check whether 49C will have any effect on the processing of the GRAS16 or in any functioning of As5. To do so, we compare the processing of the GRAS16 by a Western blot. As you can see here in the Y-type scenario, GRAS16, the substrate of the As5 is processed from its probe to mature form. If you knock down the As5 or in the knockdown parasite, you see that this processing is not taking place anymore. You see only a single band. And if you treat with 49C, it's basically similar to the wild-type scenario, suggesting that the processing of the GRAS16 is not affected in presence of the 49C. Again, to give you some idea about what actually happened, if you treat it with DMSO, you see that here the GRAS16 is labeled with green and the parasite in red, and the host nucleus is in blue. So basically, you can localize this GRAS16 protein within the host nucleus here. You can clearly see that the GRAS16 protein moving into the host nucleus. If you treat with the 49C, then you also see that there is no effect on the translocation of the GRAS16 into the host nucleus. However, in case of As5 knockout parasite, you see that the GRAS16 is not moving into the host nucleus. You don't really see the GRAS16 going into the host nucleus. This clearly suggests that 49C have a specificity for As3 like it have for plasmopsin 9 and plasmopsin 10, and it doesn't affect closely similar aspartyl proteases like plasmopsin 5 or As5 in Plasmodium and Toxoplasma. This forces us to ask the question, what is the reason behind this selectivity, or what is the basis of this molecular selectivity? In other words, why 49C can specifically target a particular class of aspartyl protease that is involved in the invasion and the egress of this parasite, but have no effect on similar other aspartyl protease. So we try to answer this question with the help of a certain tool like structural modeling, generation of the transgenic parasite, and development of the in vitro assays. I will go through them slowly one by one. So what is the reason of the specificity? For this, we firstly do a homology modeling. So basically what we did is we superimpose the structure of the As3, plasmopsin 9 and plasmopsin 10 over each other. And as you can see, if you superimpose the structure of this protein, the catalytics, the flap and the flexible loop. So normally what happens is this aspartyl protease has the active site here. Any substrate which comes through this groove and it can process the substrate. Now you can see the flap and the flexible region of all these aspartyl protease can overlap over each other, suggesting that they have basically similar in the structure of the flap and the flexible loop. And if you dock 49C within the region, you will see that basically two phenylalanine residue, one at position 344 and one at position 386 are the two main residues which provide the selectivity of this aspartyl protease, or which is mainly responsible for the selectivity of this aspartyl protease towards this particular compound. Now, if we look into the other aspartyl proteases, you will see that this phenylalanine residue, which came up in our structural modeling, is conserved in case of As3, plasmopsin 9, and plasmopsin 10. Whereas in case of the other aspartyl protease like As5 or plasmopsin 5, it is replaced with a tyrosine instead of phenylalanine residue. So we thought that if we mutate these phenylalanine residues to tyrosine residues, we can generate the aspartyl proteases form of plasmopsin 9 and As3, which will not be sensitive to 49C anymore. And we did that by developing a large number of ways, or we addressed this by generating a large number of experiments in a different kind of way. But I am going to show you the simplest and the most convenient one. So what we did with the help of the CRISPR-Cas9 and the guide RNA, we specifically introduce a point mutation in the TG-AS3 locus. So basically what we did is we changed the sequence of the wild type AS3 in the phenylalanine position with an artificial tyrosine by providing it from the outside, thus resulting in a generation of a transgenic parasite where AS3 have a tyrosine residue instead of the phenylalanine residue. Okay, so when we look into this mutant parasite to see how this parasite is behaving in presence of 49C, now as you can see here, in case of the wild type parasite, if you add 49C in 100 nanomolar or 500 nanomolar concentration, you see that this enzyme, aspartyl protease 3, you see a starting accumulation of the mature form of the enzyme. However, in case of the mutant parasite, you really don't see this at the 100 nanomolar. However, if you increase the concentration of the drug to 500 nanomolar, you again see the accumulation of the mature form. So what actually is happening is, if you see that the pro form, there is hardly any difference. So when the pro form of the enzyme is present, or when the full length enzyme is present, which is 70 kilodalton, the pro region is covering the flap region of this enzyme, which is basically preventing this compound from interacting with the flap region. However, when the protein is processed from its 70 to 55 kilodalton, roughly here, you can see this compound can clearly come and bind, interact with the flap region of the enzyme, which thus results in the stability of this enzyme in case of the wild type parasite. However, if we look into the mutant parasite, you see that in the mature form, at least in 100 nanomolar concentration, this accumulation of the mature form of the AS3 or the stabilization effect of the 49C is much less, suggesting that there is probably the mutation is affecting or hindering the 49C to interact with the mature form of the enzyme. However, if you put in more compound, you can see that enzyme is still susceptible to the 49C. So finally, what we check is what is really the effect of this interaction of the compound with the processing of the microneme and the log-tree protein. As you can see here in the same similar processing assay with the microneme protein that I have shown you before. In the wild-type parasite, you treat with 49C, you started seeing an accumulation of unprocessed microneme protein, and MYC6 is again used as an example here. And in the mutant parasite, where you have the phenylalanine to tyrosine mutation, you see similar to 100 nanomolar, where there is no basically reduced interaction of the aspartyl protease of the compound with aspartyl protease 3. You see that there is no such accumulation of the mature form of MYC6. However, again, you increase the concentration of the drug, you started seeing an accumulation of the MYC6. So we quantify what is the actual result, what is the actual, the numerical value of this increased resistance. And we see that in vivo, if wild type parasite have an IC50 of 0.66 nanomolar to the 49C, which is roughly increased threefold in presence of the mutation. However, as you can clearly see for yourself, the compound is not completely ineffective even in the presence of the mutation. So basically, we thought that our mutation is not optimal, since normally if you do a mutation and develop a resistance for this drug in the intracellular protozoan pathogen, you sometimes expect to see at least 100-fold increase in the 100-fold increase in the IC50. We are not getting that. So we decided to follow an unbiased approach, so that we thought that let's nature give us what is the reason for this, or what is the bias for this resistance or the susceptibility to the particular drug. So what we did was an unbiased approach. So we applied a process which is called the chemical mutagenesis. So, with the help of the mutagen agent, like here we use EMS, which are using mutation, which will induce mutation in the genome of the parasite. So we culture the parasite in the presence of this compound, and after certain passages, we started to select this parasite in presence of the IC50 5x or 2x concentration of the 49C, in the hope that as a result of this random mutation, some of the parasite will have some genetic changes that will make them resistant to 49C. However, unfortunately, when we test with the 5x concentration of the IC50, all our parasite died. So the population crashed and we were not able to recover any single mutant clone. However, when we used the 2x concentration of the IC50 of the drug, we see some colonies appearing. And when we took this parasite and isolate the ASP3 gene and sequence the ASP3 gene for this mutant parasite, we see that the same phenylalanine residue in the position 344 is mutated in all these resistant parasites. So basically they have been mutated from phenylalanine to cysteine, unlike the tyrosine residue that we are getting. However, in terms of IC50, they have roughly similar IC50, like 2 points, like roughly threefold higher than the wild type parasite. So apparently this suggests that although we go for an unbiased approach, there is no apparent increase in terms of the IC50 towards 49C. Then why did parasite selected cysteine in place of tyrosine is still a matter of doubt, and we can only hypothesize that, as I have told you before, that this aspartyl protease is a important enzyme for processing large number of substrate for these parasites, which are basically involved in the stage of invasion or egress to the host, egress out of the host. So probably the presence of the cysteine, which is a smaller group, probably accommodate the accumulation of a large number of substrate, or probably it allows the entry of the large number of substrate in the groove. So this is probably the optimal condition where the parasite can still develop resistance to 49C, yet can accommodate the maximum number of substrate as compared to tyrosine. So probably this is the reason, although this is just a hypothesis, probably this is the hypothesis that is why the parasite favored the cysteine mutation over tyrosine mutation. We tried a similar approach in Plasmodium. So 3D7 is the wild type strain. And then we also tried in W2 and DD2 strain. And we started selecting them in presence of 49C. So in presence of other drugs were used as control, like chloroquine, pyrimethamine. And you can see that for all these drugs, the 3D7 started to show that the resistant population started to appear in 9 weeks, 8 weeks, 12 weeks respectively. But 49C, with respect to 49C, we cannot really isolate a mutant clone, probably because 49C have dual target in Plasmodium, both Plasmodium 9 and 10. That is the reason probably we failed to generate any mutant line in case of the Plasmodium. So as this work was going on, we were also interested in quantifying the IC50 of several compounds in an in vitro assay system, or we wanted to develop an in vitro assay system so that we can compare compounds similar to 49C, and we don't need to infect, we can do that in vitro. So what we did is we designed a freight-based peptide-based freight assay. So as you know, this ASP3 is already a substrate. ASP3 is already a protease that cleaves several proteins of micronins and ROP3. So we have a rough idea about the substrate of this aspartyl protease and where it cleaves its substrate. So what we did, we attached a fluorophore and a quencher on the either side of the peptide, substrate peptide, which is roughly 12 to 14 amino acid long. So this is a very short distance. So the quencher will quench the fluorescence, and when the peptide is intact, you will not see any fluorescence. However, if you produce the recombinant protein, and if your protease is able to cleave the peptide, then the fluorescence will move away from the quencher and you will see the fluorescence appearing. By using this, we were able to generate or screen a large number of inhibitory library against the 49C, and we developed this assay for the Pfizer basically to test a library of inhibitors that it tested against the plasmapsin. And as you can see here, this is also true, and this also assays also give us about the substrate's idea about the substrate specificity of this aspartate protease. For instance, you can see that the PF-RAP1, which can be cleaved by plasmapsin 9, is not cleaved by plasmapsin 10. On the other hand, PF sub 1, which is not cleaved by plasmapsin 9, can be cleaved by plasmapsin 10. So this clearly talks about the differential substrate specificity of these two enzymes in the Plasmodium, clearly suggesting that one of them might be responsible for processing the ROP3 protein, that we have seen previously for the entry of the parasite into the host, where other might be responsible for the processing of those proteins which are largely involved in the egress or the exit of this parasite from the host. So basically, if I sum up my data, so what we see that 49C specifically target a particular region of this aspartyl protease, and 49C have different selectivity between the Toxoplasma aspartyl protease and Plasmodium aspartyl protease. And despite this differential substrate specificity, each of these enzymes are able to map the critical structural unit, that is this phenylalanine position in the flapped region, which can make them susceptible to this particular class of compound. So with this, I will finish the presentation, and I will like to acknowledge. So mostly the data I have of my PhD were not shown here. So I did, as you know, that I did my PhD from CESA at ISCB, and this is Dominic's lab, and this is University of Geneva, and this was my office. And most of the work that I talked today was done during my postdoc years in Dominic's lab. So with this, I will thank you all for listening, and stay safe. And if you have any question, I will be happy to answer. Okay, so am I muted? No, I'm not.
You're okay. Okay. So thank you, Dr. Mukherjee, once again for this excellent, nice presentation. For this 1 hour long lecture was full of informations regarding updated versions of parasitology and immunology, and the possible way of drug designing against a particular parasitic enzymes. And we have few questions for you from the end of participants. May I request Professor Papia Ghosh to take care of these questions and deliver it to Dr. Mukherjee for answering, because only 4 or 5 questions are there.
Okay, so you know this 49C 1 nanomolar or 10 nanomolar concentration. Okay, so this compound, as I have talked to you, that these were product of the acetylene pharma. So before they did anything, they tested the pharmacokinetics of this compound in the mice. So we don't have to do that. So with the IP injection, all of this compound was bioavailable. Unfortunately, what was the problem is with the oral dose, MMV was not willing to go for anything that is not oral. So this was a problem. So certain modifications need to be done on the chemical structure of the compound to make it soluble so it can go orally. But regarding the bioavailability, it was not a problem. I think it was pretty sustained. I don't remember the exact time, but I think for 72 hours they checked and they can detect the compound successfully. Well, you have to look at the so this when this modeling was done, all the compound 49C and 49B was docked into, and thermodynamic calculation like delta G negative was done. So,
Okay.
so I cannot. I mean, if you want to look into the chemical nature of this interaction, you see that this docking of this compound within the aspartyl protease structure always results in a higher delta G compared to tyrosine mutation. So that is why probably when you do a mutation, the flap orientation is completely changing. So you remember the groove I shown you, so that the compound can access the active site of the compound. It's probably hindering the flexibility of the flap. So what we understand from the modeling people, that when you change the when you mutate it from phenylalanine to tyrosine, it's making the movement of the flap over this cavity, or over this a cavity of the aspartate protease where the active site is present, is much more slower. So the flap is not opening, it remains closed. That is why it's affecting the susceptibility towards the compound. So basically, we were not able to have a mutation. To answer your question honestly, we are not able to have a mutation that will completely abolish the 49C effect on the aspartyl protease. So as I have shown in my presentation, like in lower nanomolar, you see an increase in resistivity towards the compound. But if you increase the concentration of the drug, you see that this is not reflecting. So what we can say that this is such an important enzyme if you try to completely mutate it, because we even cannot generate the clean knockout for this enzyme. So I don't think the parasite would have survived. So we will not have any clone. So probably it choose for an optimum condition where it can develop some amount of resistance towards 49C and still accommodate large number of substrate which it can process for the functionality or the life cycle of these parasites. Okay, so it's a myeloid differentiation adapter protein. So basically, if you see the TLS signaling, you will see that TLS signaling can occur either on myoid dependent or independent way. So I have not shown the independent way path. So when we are checking the signaling with respect to antimony resistant parasite infection, how it can modulate the host cell, we saw that when we do the infection in the immediate knockout animal, it has a severe effect, whether as in the TRIF or the other component that is not immediately dependent, that has no effect. So it's basically adapter protein which is present in the downstream of the TLS signaling and is necessary for the TLS signaling. So imagine invasion is blocked. So let me talk about this in a detail. So what happens is when the Plasmodium parasite are dropped in your skin from a mosquito bite, they initially are in the skin cells, but they reach the liver. Okay, when they reach the liver, first they encountered is the Kupffer cells, but this parasite does not grow or replicate in the Kupffer cells. They straight pass through the Kupffer cell, ultimately making their way to the hepatocytes. When they reach the hepatocytes, only then they grow and replicate. So you can imagine anything that have the initial invasion block, so it will not be able to reach in the liver, and the parasite will get cleared from the host by immune system. So this is what will happen. Well, they recognize it, but like any intracellular protozoan pathogen, they have different strategies to make them invisible to the host immune system. So there are different ways Leishmania parasite can do it. Even as a matter of fact, the drug-sensitive Leishmania parasite also do it. So what they does it, they basically affect the process of antigen presentation. So you see that there are certain signaling molecules. So in order for the antigen presentation to happen, you need to process the proteins of the parasite. So they affect this processing machinery. again, they can affect the binding of the or the loading of this peptide to the MHC complex. So basically, they have the conformational change in the MHC so that it doesn't bind to the peptide anymore. And even if the binding happen, they can mimic the membrane fluid. Basically, they quench cholesterol from the host membrane. So the peptide MHC may reach the host cell membrane in the intact configuration. Yet, considering the fluidity of the membrane, they cannot generate the proper TCS signaling. And this is not unique only for Leishmania. This is also true for several other intracellular protozoan pathogen, be it Toxoplasma or trypanosomes. That is how they normally escape the immune system. Yeah, I think I have already answered this if I have understood the question correctly. So you were saying that why the signal appear firstly on the liver. Okay, the parasite firstly colonize on the liver. So to answer your question, the blue color appear in the liver as the parasite after their infection goes directly into the liver hepatocytes, and then they at certain point they try to invade or their asexual stage comes up, and then they go into the blood. So as soon as they started to leave the liver, the signal in the liver started disappearing, and you can see the signal coming in the blood cells. However, if there is a defect in the invasion, like in the presence of 49C, they will be not be able to enter into the blood cell or infect the blood cells. So gradually they will be cleared by the immune system. So that's why you don't see a continuous signal. Okay, then we are called. Yeah. Okay, thank you. So I can switch off, right? Okay, thank. Okay, okay. Yeah. Okay.
Yeah. Yeah, welcome Mainak. Good evening. So I'm happy to introduce Dr. Mainak Sengupta, my colleagues at University of Calcutta and from the Department of Genetics. Dr. Sengupta completed his PhD in life science from Jadavpur University. And he has more than 33 international publications in the field of human genetics. Dr. Mainak Sengupta reported for the first time ever 11 new human mutations related to human diseases. He has received 7 major research grants for conducting research in the area of human genetics. He has prestigious professional memberships in the different scientific societies. He is life member and assistant secretary of Calcutta Consortiums of Human Genetics, life member of Indian Society of Human Genetics, life member of Indian Science Congress Association, life member of Indian Academy of Neurosciences. He is reviewer and the member of editorial board of many reputed international journals like BMC, PLOS, Genetics, and guest editor of Journal of Ophthalmology. He has received several prestigious awards and fellowship like Tulsabai Somani Educational Trust Young Scientist Award in 2018, Rabindranath Tagore Research Grant Award for Promotion of Science, Education and Research by West Bengal State Council of Science and Technology, Government of West Bengal, India, ISHG Young Scientist Award 2012 in the 37th Annual Conference of Indian Society of the Human Genetics in the year 2012 at Chandigarh, India. And he has many credentials. Mainak is very popular teacher. He's a good teacher, and I think today's student will be enriched regarding the latest updates of certain molecular techniques that are usually carried in our laboratory in day-to-day analysis of genetics and molecular biology. So now it's over to Mainak, and Mainak, please start your deliberation.
Thank you, Sujata, for your nice introduction. So first and foremost, I would like to thank the organizers for inviting me to have a discussion, I would say discussion, not a presentation, with the undergraduate students and the postgraduate students. So for the last hour, I think, I was listening to my IICB compatriot Budhaditya, his lecture. My lecture would be completely different from his, which was wonderful, informative, and data oriented. I would like to focus more on the fundamentals of biology, and in a sort of teaching mode, I will skip from the teaching to my research and then go back to teaching, etc. So without further ado, I would like to, Sujata, please see that whether or not I'll be able to share the screen. Yeah, but it's not coming. Is it coming? Okay. Just a second.
Mainak, open the PowerPoint first,
Yeah,
then open the Zoom screen.
Right. Right. Did that? Okay. Oh, thank you. So thank you for the help. So as my um, title reads, it's genomics and its impact on health and society. I would be dealing with a few like unexplored aspects of genomics to make sure uh, to tell the students that genomics and genetics is not all about cancer diseases, cancer related diseases, like to study the mutational aspects of diseases, but it is much more than that. Genomics is much more than disease. And to start with, thereby, I would show something with respect to mountaineering, with which I am conversant with. I like climbing, I like hiking. So this peak that you are seeing is called as Meru Peak. It is present in Uttarakhand, and it is a very hard peak to climb. Uh, these three gentlemen, they tried to climb the Meru Peak, as it is written over here, the mountain Meru, located near the headwaters of Ganges in northern India. It is about 22,000 foot peak, but the last 1,500 foot of this is a smooth granite face called the shark's fin. Because of that smooth face, it was absolutely impossible to climb. These three fine gentlemen, Konrad Anker, Jimmy Chin, and Renan Osturk, I don't expect you to remember the names, but these three individuals wanted to climb the peak. But when they were very near the peak, then the weather became absolutely murky and they had to come back. That was a heartbreak for them. But they were preparing to go to that peak again. But within a year, a disaster struck. In Swiss Alps, Renan Ostood faced with an avalanche, and he was almost killed. He had like broken vertebra, his ribs were shattered, and thus was the dream of conquering this Meru peak again, because they were to climb, they were to have this adventure of like reconquering the Meru peak by next 3 years. So it was almost shattered, or next 2 years, I think. However, within next 2 years, these three individuals actually, this is Renan Ostood in a hospital, almost in the deathbed. But what happened? It was a miracle, and this is Renan Ostood standing over the Meru peak 2 years after the fatal accident. Doctors told him that you cannot go, you are risking your life. It is too much, it is not worth this, a risk that you are taking. Well, you can climb that particular peak, but you will be dead. But he didn't listen to them. He suffered a minor concussion on the way, but he reached the summit. That was the first civilian to reach the summit of Meru Peak. Why am I showing this in a lecture about genetics and genomics? The question is, why did he take the risk? Not only him, why do any of us take a risk? Like this person is doing what we know as bungee jumping. Although there is a rope attached, it is an extremely risky proposition. But this guy is doing it. And we know a lot of individuals who do it. Not only these things, when suppose in a class is going on, a certain student during the class is sort of chatting, is doing WhatsApp in a mobile. He or she is also taking a risk. Now, why would individuals like to take the risk? I would like all the students and everyone listening to it to remember this question. We will return with this question at the very fag end of my talk. Now, when I was a kid, questions like this always used to appear, and I used to bother my father very much by asking that why is it like that, why it is like that. My father, being a non-biologist kind of a person, would answer to a little of it, but not more than that. One of them was, why are we all different from each other? I used to stand on a road on my father's lap, and I used to say that, okay, this person has beard, this person does not have beard, this person is a little fairer, this person does have a different kind of a eyebrow. Why? So from the very childhood, I used to ask that, why are individuals so very different from each other? So in a crowd, every individual is actually different from each other. So some are quite like ravishingly beautiful, some are almost sharp like anything, very intelligent, some are dumb, some are a good fighter, some have blue eyes, etc., etc., etc. But they are not similar. So why they are not similar? So is it because of the environmental effect? Is it because of the climate where they are growing up? Is it because of the differential growing up status? That is the nutritional difference, the educational difference, etc., 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 which is making these individuals so very different from each other. Well, if that is the answer, then what about them? These are all the brothers, but yet you can also see that there is a huge amount of difference between them. So let us ask ourselves a very pertinent question. Why and how are normal humans like us, like all the individuals who are present in this webinar, are different with respect to features like color, height, etc., etc., etc.? Let's go to the next layer of question. Now this slide shows diseased individuals or individuals with certain syndromes, certain disorders. As you can see, this individual is having albino. This is his brother who is not an albino. You know that this is an extra finger, this is progeria, this is hypertrichosis, there is a lot of hairs in the ears. Now let us ask the second question. Why and how are normal human beings different from diseased human beings? So what is wrong? Both are human. So why are they different? Now we know that because of the genes might be the answer. Genes are responsible for the inherited characteristics that distinguish one individual from another. Now, what is the gene doing? The genes are not doing nothing but producing proteins. This is just very oversimplified answer, but still now we see that the like the genetics books has on this definition, that is, genes are the coding region of the DNA which gives rise to proteins. Gene is much more than that. We will discuss it later on. So the proteins are the building blocks of the human body. We all know that. The protein is coming from mRNA. The mRNA is being transcribed from the DNA, and a little part of the DNA is the gene. It is coding for a functional mRNA or a functional protein. Okay, and genes are responsible for each and every parameter of each and every characteristic feature of a humankind, of a human being, like the immune response, the heart rate, behavior, movement, sex determination, blood clotting, cognition, cell cycle. Name a trait, and I will say that there is a gene behind it. Okay, immune response, you can very well understand. You now know that two individuals with two different kinds of immune response might not be equally susceptible to COVID-19. We now are very conversant with this particular phenomenon. So now let's, with this idea, let's go back to these two questions. Why and how are normal humans different with respect to features, color, height, etc.? And why and how are normal individuals different from a diseased one? Is it because the genes are different? That is, I do have gene A, the other person does have gene B? No. I also have gene A, he or she does also have gene B. What is different is the genetic variations. The nucleotide sequences which make up the genes might not be uniform across human population, across plant population, across animal population. So we know that like the nucleotides means, or there are only 4 nucleotides. It's the combination of these 4 nucleotides that make up the entire human genome. For that matter, genome of any organism. They are adenine, thymine, guanine, cytosine. What is different from individual to individual is these specific combinations. Suppose in a specific location, let us name it as 100. 100th position, there is adenine in a specific individual, there is thymine or guanosine in another individual. Because of this one change, something in the protein might alter. Because of that altered protein, the look, the height, the intelligence of that particular individual might differ from that of the other individual. And this signature, the nucleotide sequences, this is the variable parameter between individual to individual. So if you study the individuals, you will see there these yellow markings are the different marks. This is a random example. If you take randomly 50 individuals and you study a specific region of the genome, you will see there are lots of variations within them. But the genes that they have is exactly the same number of genes. So it is the genetic variations that are different. But this is still not answering these two questions, that if the genetic variations are different, then why one sort of genetic variations are creating differences in our height, color, etc., and why some other genetic variations are making individuals diseased. So this is bizarre, that genetic variation is causing an individual to be diseased. Why should it be like that? So for that, let's take an example. This happens because of two particular types of genetic variations called as mutations and polymorphisms. So what are mutations and what are polymorphisms? Let me read out from one of the books. This is a bookish definition of mutation and polymorphism. So the nucleotide variations that stay in a population in a frequency of more than 1% are polymorphisms, and those that are lesser are mutations. Now, come on, you can say that this is why should I believe this? This is so very statistical a definition. I mean, why should some genetic variations which are like beyond 1%, I would call them polymorphism, and less than 1% are mutations? Why is it like that? Let's try to put it in a more functional way, in a more cartoon way, in a sort of more lucid way. So suppose that these two skeletons are two sibs, two brothers or two sisters of family one. You can see very well that there is one particular change here. This ATGC, this is a random part of the DNA sequence. This ATGC is not exactly similar to this here you can find that instead of C, this brother or sister does have T. But this particular T is such a variant which is not causing any problem whatsoever in the protein that it is producing. The protein is remaining just fine. So this individual is just as fit as this individual. Thereby, this individual can reproduce with as much efficiency as this individual. Now, after 1,000 years, if you are given a task to find out what will be the frequency of T, because remember, this individual had passed on this particular T to the next generation with equal proficiency as this individual. So now suppose you have made a survey and you found that among 100 individuals, 40 individuals carry T. So thereby you can find out that this T thereby is having a frequency of more than 1%. We name such a genetic variation as a polymorphism. So this is much like two footballs with two different panels. So maybe this individual, because of this C, this individual is a bit fairer. Because of this T, this individual is a bit darker. So it is but none are diseased. You can play the football with both the footballs, just that the structure is different. The structure is polymorphic. As opposed to that, you consider another family where there is another change. The change is in another position. This C is T here. Because of this, what happens is the protein is totally malfunctional. The protein that it was being produced is totally malfunctional here. Then what will happen? It will be like the football and a punctured football. Thereby, this individual will lose the efficiency of the health will suffer. This individual will probably not be able to propagate, will not be able to reproduce as efficiently as him or her. Thereby, what will happen? Maybe he or she will die early, and ultimately then, if you are given the same exercise as this, that is after 1,000 years, you have been told to find out the frequency of T. But remember, this particular individual died before he or she could reproduce. So this T died with him or her. Thus, the frequency of this T will become 0. That is less than 1% we are considering. Thus, this type of a variation, which because of deselection by death, deselection by reproductive inability is becoming lower and lower in a society, we call these types of changes as mutations. So this is the basic difference between a polymorphism, which you will be finding in general population more than 1%, and a mutation, which you will be finding lesser and lesser and lesser in a population, because they are deleterious. They are not allowing the individual to survive and reproduce as efficiently as the normal individual does. And this actually was the basis of, so we can say polymorphism stays in population in spite of being different from that of the normal. Mutations, the nature is acting against it. That is as if the nature is deselecting, is taking out, is excluding that particular variation from the genome. This was the basis of, although he did not know about these genes and things like that, but this was the very basis, the molecular basis of the idea of this great man, survival of the fittest and natural selection. That is, those individuals who will have genetic variations which will push them to be either better or similar, they will survive. Those genetic variations which are making an individual, it can be an animal, it can be a plant, it can be a human, those genetic variations which are making that particular human being or animal or plant lesser adaptable, and thereby they are not being able to reproduce properly. Thus, they are eliminated. With their elimination, the variation which actually pushed this particular individual to be diseased is also being eliminated. This is the theory, molecular theory of natural selection or survival of the fittest. So we know that thus genes shape everything about us and our variations. Then it was time to know the entire content of the genome. We all know that the Human Genome Project, one of the greatest projects of our time, was conceived, and there were a lot of project goals. The ultimate goal was to identify the approximately 20,000 to 25,000 genes in the human DNA, determine the sequences of the entire genome that we have, store this information in database, improve tools for data analysis, how to analyze this particular huge set of data, etc., etc. These are the two gentlemen who independently got us our human genome database, our human genome project. The human genome was sequenced. Craig Venter, the person at the left is Craig Venter, the person with specs is Francis Collins. So there was two different projects parallelly going on, which almost at the same time, they inferred that yes, the whole human genome is solved. And this is the library of human genome. You can see that there are a lot of, these are books with chromosome numbers written over it, 13, 14, 18, Y, X. You open the book, this is all about ATGC. Just line after line, ATGC is written. So the first letter is the first base of P arm of that particular chromosome, and the last letter in that book is the last base of the Q arm of that particular chromosome. Isn't that fascinating? So the revelations of Human Genome Project in a nutshell was they could sequence more than 99% of the human genome. They found genes that code for diseases and inherited conditions. So that was what they were looking for, genes and diseases of the inherited conditions. They found why some people have red hair and some blonde. So I will stop here. This particular thing, they found why some people have red hair and some blonde. This intrigued me. This always had appealed me to look into the profile. Why would the coloration, why would the skin tone of individuals be so very different? And India is a wonderful country to look into the human pigmentation variation. So let's talk about human pigmentation variation, which is my own work. I did my PhD thesis on human pigmentation variation. One of the projects that is still running in my lab is on human pigmentation variation. So this particular scale, this particular map is called as von Luskin scale of human pigmentation variation. So what they used to do before the spectrophotometric camera was discovered, they used to take glasses of different shades. Those glasses were put on the unexposed area of individuals, like the upper arm, etc., etc. And they used to see that which glass was corresponding to the skin tone of that particular individual. And they made this entire map. You can see that the Indian map already does have a lot of variations of these. So again, I would not expect you, any of you, this is not an exam-oriented teaching at all. So I do not expect you to remember the names of the genes. That is not important. But I just want you to know that what was revealed from our research and many others' research was polymorphisms. We have already known that what polymorphisms are. Polymorphisms are genes like tyrosinase, SLC45A2, etc., etc. These variations are the responsible variations because of which the South Asian populations, some individuals are very dark, some individuals are very fair. Even in South India, in Kerala, you can find a lot of individuals who are very fair and a lot of individuals who are very dark. This is because of genetic variations in these three genes. Okay, so this is the so very difference. You can see Vietnamese, Thai, Cambodians. In Cambodia, there is a very fair individual and a brownish individual. Similarly, the blue-brown eye color. This can also be explained by variations in a gene called as OCA2. There's so many varieties of eye color, and all the genes are known whose variations are responsible for these different eye colors. Similarly, among the East Asians, whom we call as Mongoloids, they are fair, but their fairness is completely different with that of the European fairness or the Latin American fairness. The Mongoloid fairness is a bit of yellow. So it has been found that again, a polymorphism in a gene called as OCA2, if it is present, it makes the fairness, it makes the pigmentation a bit yellowish. Those who have read Sherlock Holmes, the red-headed league, the red hair phenotype was found to be because of an allele called as MC1R variant. So by this, you can very well understand that this many number of genes and their variations are responsible for the human pigmentation variation across the globe. Very interestingly, I have typed a few genes names with bolded green. in the fag end of the slide, it was found that if you, by siRNA treatment or by like deletion, if you just delete, if you just nullify these particular genes, that is, if these genes are not present in the body, then what happens is a phenomenon called as albinism, where pigmentation is not present. Till now, we were saying that polymorphisms were causing the different shades of variations between a fair and a darker individual. Now comes disease. So mutations in these genes cause a disease called as albinism, where there is no pigmentation. Why is albinism a disease? Because pigmentation is needed for proper eye development. Pigmentation at postcoitus 13.5, that is 13.5 days after fertilization, if there is no pigmentation present, if there is no melanin present, then our eye development suffers. We become legally blind. Albinism individuals see like this. They have to read like this. They cannot open their eyes in the sunny environment. Thereby, it's a disease. And I work with the genomics of albinism. So suppose now that we know we have extracted the cell out of a patient, we have extracted out the DNA from the blood, we knew the sequence, and then we found out that, okay, this particular individual who is obese, who is an albino, does have this particular genetic mutation. Then what? What should we do? We have found the faulty position, but then what is it that we can do? So before going into that, what is it thing, we want to know that how can we identify this particular genetic change? So that is called as gene testing. And this gene testing is done by two very basic procedures, which I'm sure that you have heard of it. One is PCR, polymerase chain reaction, and another is sequencing. So let's go and like study and like tell ourselves that whether or not we have been able to understand this gene testing phenomenon. Because until and unless we do, we will not be able to move forward to the next more complex slides. So what is polymerase chain reaction? Polymerase chain reaction means a reaction which is being done by DNA polymerase. As simple as that. So this is a particular DNA, two DNA strands. One is 5' to 3', another is 3' to 5'. So these two DNA strands need to be replicated. Polymerase chain reaction is nothing but in vitro replication. That whatever the replication is being done in our body, you are trying to simulate it in a test tube. That is polymerase chain reaction. So this is, I mean, to make it more easy, I can say this is about like xeroxing 2 pages of a book. So these may be 2 pages of a note. We always tend to xerox notes, so probably that would be more familiar. So it's about xeroxing 2 pages. But you know that the person who is xeroxing, whenever you give him him or her the notes, he will first unclip it. So the unclipping of the 2 pages is essential. And unclipping of the 2 strands of DNA is also essential for PCR, because the 2 strands are like through hydrogen bonding, they're bound to each other. So how to unclip? Just add heat. Heat the DNA and the DNA will denature. So this step is called as denaturation. It is normally done at 94 degrees centigrade. So after this, what happens is it's easy. Then take the DNA polymerase and the new strands will develop. However, it's not that easy. DNA polymerase cannot work alone. He always says that I need a helper. I need someone who will help me to like place myself on the DNA. Unlike RNA polymerase, DNA polymerase cannot do this thing alone. So what does it need? Tell me, what do we need when we like color our house? Do we apply the color at the very first go? No, we apply a primer. Then comes the color. Exactly similarly, DNA polymerase also needs a primer. Primer is a little complementary sequence in which will sit here, which will also sit here in the 2 different strands. So the primers will first come and sit, and then the DNA polymerase will work. Now this sitting of the primers over the denatured strands of DNA is called as annealing. So we have known about the denaturation. Now we are like going through annealing. Okay, fine. It is annealed. Then the work is easy. The DNA polymerase will move. There will be a lot of nucleotides. The DNA polymerase will polymerize each nucleotide. Of course, the nucleotides are not poured like this as in the picture in drums, but it is required in very minute quantity. It is done by pipette man. So the DNA polymerase incorporates the nucleotides considering the complementarity of the bases in the template strand, and gradually the entire DNA strand is amplified. Okay, so this part is called as extension. So this denaturation, which is done at 94 degrees centigrade, annealing temperature varies between primer to primer. Extension is generally at 72 degrees centigrade because the polymerase, the Taq polymerase that we work on with in case of PCR, the activity temperature is about 72 degrees centigrade. Now this is a cycle. That is, the DNA needs to be denatured, the primers needs to sit, and then the polymerase needs to polymerize. It needs to extend. This entire cycle is repeated time after time after time, maybe 30 cycles, maybe 35 cycles, maybe 40 cycles. Thus, the polymerase reaction gets the name polymerase chain reaction. So these three continuously in a chain are repeated till we get a lot of DNA like this covering the entirety of my slide. So this is how PCR occurs. Okay, now fine. You have done the PCR. Then whether or not the PCR passed or failed can be quickly checked in gel electrophoresis. I'm sure that everyone knows about gel electrophoresis. You just need to pour the DNA. You just need to subject the DNA from a negative charge to positive charge. As we know that the negative charge DNA are negative charge, so it will move towards the positive charge, and in doing so, it will give bands. So bands like this in a agarose gel electrophoresis system, it will prove that, okay, you have actually been able to amplify the DNA. Fair enough. But have we been able to read what is the content of this DNA? No. We have been able to just identify, we have been able to just amplify the DNA, not read it. So PCR helps us to amplify the DNA, but reading is something else. How can we read what is the content of the DNA? Suppose this is the content of the template. How can we read it? For reading it also, we need a little part of the sequence that is already known. So suppose the yellow marked one, like ATG, CATG, this is already known. Now the green marked one is the green marked part is the one that we want to read. We want to know what is the sequence of this. We have got no idea. So what to do? We will be taking the help of the similar PCR mechanism to identify, to read these sequences. Okay. So first, let's go back to this drum of nucleotides thing. Now for sequencing, for reading this particular sequence, what we need to do, we need to alter a little of the content of this drum of nucleotides. What will we do? We will deliberately add, along with the 4 nucleotides, we will deliberately add a few modified nucleotides. So what sort of modification? The modified nucleotides will stop extension. So wherever the modified nucleotide will sit, it will stop the polymerase in further extending. And to add twist to the tail, these particular modified nucleotides are colored. They have been given specific fluorophore, like adenine has been given green fluorophore, thymine has been given red fluorophore, G black and C blue. So they are marked. So wherever these modified nucleotides are coming and sitting, you can understand that, okay, so G has sit, but it will not extend. It will stop the extension. Let's take an example. So suppose this is a primer. You can see that the primer will be complementary. I'm sure that every one of you knows about complementarity. It will be complementary to the known sequence. Now you have added the nucleotides and the polymerase. So suppose this is C. So a G would come and sit here. So a polymerase is adding a G. So suppose this G is that modified nucleotide. Then what will happen? Immediately there will be no further extension and the sequencing will be terminated. Thus, the sequence is terminated at on that G only because this G is modified. Well, you can understand, you know that, okay, G has been added. So you have read one base. But you have to read further 16 bases, further 17 bases. If the sequence is terminated, how will you read? Now let's go back to PCR again. Remember this slide? So we do not have with us only one fragment of DNA. Remember, we have amplified that. That means one particular fragment has been amplified 1,000 times. So whatever we saw that this is happening, this might happen in one of the fragments, but not in all. Maybe in another fragment, what was happening is this C, this particular C, is a normal nucleotide, not this modified nucleotide. This is by chance. A normal nucleotide may be added, a modified nucleotide may be added. In the first case, a modified nucleotide was added and it shut down the extension. Now it is a normal nucleotide that is added and it will extend. But in the second position, an abnormal, that is a modified nucleotide again was added. Now from now on, I will mark the modified nucleotide with a star. So here in the second position, a modified nucleotide was added. So it will happen. It's the same thing will happen. No more extension. In the third fragment of PCR, the C was added, a normal T was added, and the third base becomes modified. Now, what happens is, from the third base only, the extension stops. So what am I doing? By adding this modified nucleotides is, I'm actually preparing a lot of short fragments. This fragment is where C was added, the C modified nucleotide was added. This fragment is where a modified T nucleotide is added. This fragment is where a modified A nucleotide is added, and likewise. So by doing this modified nucleotide based PCR, I am generating short terminated fragments. Remember, one terminated fragment will anneal here in the very last part. So we have to assume that for all the rest of the nucleotides, normal nucleotides would have been added. In the last base, an abnormal modified nucleotide is added. So we have generated a lot of truncated products, the last base of each of which are modified, and thereby you can read by the different coloration of the last modified bases. So this is what exactly happens. Suppose this is the sequence that you are trying to read. So this is the first terminated fragment because G was the modified one with a specific color. This is the second terminated fragment with two bases with a specific color. This is the third, this is the fourth, and likewise. Now, interestingly, if you read it like this, GACT, GAA, GCT. So I'm just reading the colored terminated bases, GACT, GA, AGCT. You'll find that if you read it like this is just complementary to the sequence you wanted to read. Thus, if you find out the complementary bases, you can very well understand, you can very well sort of calculate what are the exact bases, that original bases that I wanted to read. This is the mechanism of sequencing. So as these particular fragments, all these fragments, they are now set into a gel in a capillary. Capillary is nothing but a glass tube full of gel, a very small diameter. You run them in through like the capillary. The shortest fragment will run fast, and thereby, as it is running, as this shortest fragment is running, it is hit by a laser, and the color emitted is received by a detector. So this is green, so it was hit by a laser. The color was detected by a detector, and it gave an output in the form of an A peak. We call these as peaks. Then as the second fragment passes, the T has pink coloration. It is detected by the laser and the detector, and we get a T peak. So all these peaks are coming from different fragments, but this is giving us an entirety of the read of the genomic sequence. Thereby, this is sequencing is called as sequencing by termination. It was discovered by Frederick Sanger, and thereby it is also called as Sanger sequencing or first generation sequencing. So suppose you have sequenced a diseased individual. You know that this gene is responsible. You have sequenced the gene. You found out that this is the sequence. You compared that particular sequence with a sequence, same sequence of the normal individual. You found out where is the mismatch. Bingo. You found out the mutation. the mutation was detected. This is how the geneticists, this is the fundamental work that the geneticists do, whoever works on a specific disease screening. Okay, so now into my work. I work with oculocutaneous albinism. As I said, you can see that this is a particular individual who is albino. This is his father. We sequenced a particular gene, tyrosinase, TYR, in him as well as him. So this is his sequence. This is the father's sequence. You can see that there is blue peak here. There's a red peak here. So from here, you can understand that this is what a mutation is. And very, very interestingly, this individual was located in a village called as, village populated by Tili community in Bankura. In Bankura, very near to Bankura Shodor. So this entire village had lots of albino individuals. And we sequenced all of them, and we found that all of them carry the same mutation, which was very unlikely. So we published that. So this is the pedigree of a few representative individuals from that village. We sequenced all of these gray shaded individuals for albino. We sequenced all of them, and all of them had the same mutation. So now we know that, I have got the mutation, I have published the mutation, but just knowing the mutation will not solve the problem. The ultimate problem will be solved when we'll be able to revert the mutation to a normal self. That is the process to which the geneticists are looking up to for years. And now, after a long time, because of the advent of CRISPR-Cas9 mechanism, people have been able to revert the mutations to a normal phenomena, at least in 1 or 2 cases till now. CRISPR-Cas9. I'm sure that in this webinar, someone or the other will be talking about CRISPR-Cas9. You'll be getting to know how interesting a bacterial mechanism is, which we can introduce in our own eukaryotic system and can correct our genetic differences. Okay. But there are certain diseases which are congenital, like hemophilia, like albinism. So you have no chance of, unlike glaucoma, unlike schizophrenia, these things, glaucoma and schizophrenia, occurs after a certain point of time at your age, at an old age. But hemophilia, thalassemia, these are all present from birth. And if something is present from birth, how will you be able to correct it? There, what we do as geneticists, again, this is my own work, we identify carriers in case of the recessive disease. That is, in case of the diseases where both the chromosomes need to be affected, we try to find out individuals who are carriers. Why? Because if you can identify carriers, just like the asymptomatic COVID carriers, if you can identify the carriers, you can tell them that, okay, see, you are a carrier. If you marry another carrier, then there's a 25% of chance that your next generation would be diseased. Now it's up to you, but you need to know that whom you are marrying, is he or she a carrier also? If they are, then you have to take a call. What will you do? Because there's every probability that 1/4 of the individuals will be diseased, 25% chance that the diseased individuals will pass. So we are trying very hard to identify carriers for different types of diseases. So the real advancement so far, we have sequenced many genomes, we can identify disease-causing spots, we can prevent onset of diseases, and we are approaching towards gene therapy. What's next? The next best thing is personalized medicine. That is, medicine according to individuals' genetic makeup. I'm sure that each and every one of you have known that if two individuals, even two brothers and sisters, have been drenched in the rain in this very same manner, just like Opu and Durga. Durga fell ill, fatally ill, but not his brother, not her brother. We can find the same thing, that we are exposed to all kinds of things that our parents say that don't do. We take ice creams, we play a lot in the rains, but not everyone suffers equally. And even if two individuals suffer equally, then one might be recovered just after 2 doses of Calpol, the other does not. Have you ever thought, why is such a thing happening? Those particular things led the geneticists to develop personalized medicine. So let me tell you a story. A story of a girl called as Latris. Latris is a 7-year-old girl who has been diagnosed with leukemia, a type of cancer involving the bone marrow and the blood cell. So she went to a doctor for her therapy. Her chemotherapy would have started soon. The chemotherapy, the best possible drug for chemotherapy is purinethol. It's a well-known chemotherapy drug. It incorporates itself in the rapidly dividing cancer cells and kills them. This purinethol drug. But the doctor was not entirely sure because he had records that purinethol comes with very severe, sometimes fatal side effects in certain patients. Why so? The mystery was unraveled when scientists began looking at how this particular drug, purinethol, is broken down in the body. So that means how the drug is metabolized. They discovered that there is an enzyme called as TPMT. This enzyme is responsible for breaking down and inactivating the drug purinethol. And they found that the people vary in their TPMT enzyme activity level. That is, I may have 100% TPMT enzyme activity. The other person might have 50% TPMT enzyme activity. The third person might have 1% TPMT enzyme activity. And these things are happening because of the different DNA polymorphisms of TPMT in these 3 individuals. So, because purinethol is a toxic substance, it needs to be broken down. Because if it stays in the human system for long, it does have different other side effects. Okay, so you have to give purinethol its scope to act, and then immediately when the action has already begun, then you break down the drug. That is a known parameter for all good drugs. So, but if TPMT enzymes are inactive, then the purinethol will remain in your body. In some individuals, TPMT enzyme was found to be hyperactive. That is, just as you get purinethol, TPMT destroys it. So purinethol's action remains to be served. So it was done, this study was done in America. It was found that 89% of children have actually full TPMT enzyme activity, and thus they can receive a full dose of purinethol. 11% of children have partial enzymatic activity, and thus you cannot give full purinethol dose. You have to give them a lower purinethol dose. There is 0.33% of children, they have insufficient enzymatic activity, and thereby they shouldn't be given purinethol at all. They must be given some other drug. These all are happening because of genetic variations. Now suppose this particular individual who is having only 1% of TPMT enzyme activity, he has been given full dose of purinethol. That person had come to the clinic for curing leukemia. That person would die because of the side effect of purinethol, because purinethol could not be broken down. Thus is the importance, such is the importance of personalized medicine. Such is the importance of knowing the genetic makeup of each individual and then prescribing the medicine. Is it looking far-fetched? Are you thinking that this is too much to do? No. This has already been started in countries like England. In Sussex, there is a clinical laboratory. There is a clinical, there's a series of doctors who routinely test for a specific gene, and then they apply the drugs. This is the call of the day, personalized medicine. So, the personalized medicine has to be according to individuals' genetic makeup. Now, let's go and explore a completely different posture, music. You definitely are asking that what is music to do with genes and genetics? It has. So you can get to know a lot of individuals, your friends, who completely, like a music which is completely different from your liking. So one might say that he likes rap and country. You are saying that, so basically your musical taste sucks because I like Rabindra Sangeet more. This is a very apparent phenomenon. Have you ever thought that why this type of a thing is happening? And there are even individuals who do not like music at all. There are individuals who cannot understand that if a person is singing badly because they are tone deaf. They cannot identify if two individuals are singing in two different tones. This led the geneticists to study the genetics of music, and they found out something absolutely, absolutely fascinating. Before going into their genetic discovery, I would urge each and everyone to go and to download this particular documentary, Alive Inside, and just see what is memory, what is music in terms of memory, how music is intertwined inside us. Actually, the musical cadences, the musical ability of human beings preceded language. If it is, you are considering that this is too much of a bluff, it cannot happen. No. First, individuals learned to play musical instruments earlier than language developed. So music is hardwired into our system. This Alive Inside documentary is about a doctor who visits tertiary centers of dementia, of other neurological diseases, where a person has completely become vegetative. All his or her memories are lost. All this doctor used to do was to pass on an iPad to the individuals. They get to know information. He first talked with the family members. He wanted to know what sort of music he or she loved to hear. Before this type of thing happened, he used to load the iPad with those music, with those songs, and gave them these iPads. This is such an individual who was vegetative for last 7 years. He does not say anything to whatever asked. He just sits like this, just like this, with a cynical face. This is his granddaughter who always comes and tries to feed him, but he just does not recognize him. So this doctor goes and gives him an iPad. Just see the change of the facial expression of this individual who is completely vegetative for the last 7 years. See, here he's crying. And you will also cry if you see the entire episode of this Alive Inside documentary. All I'm trying to say is how music is bringing back memories. How is it able to do? It's an external parameter. Because music acts like a drug. Just like a drug, a ligand acts, music changes itself to certain chemicals which hits to unknown receptors and functions like this. So, and here this guy is crying and he's saying things for the very first time in 7 years. Is thereby music in our genes? There was fascinating studies, as I said, going on, and people found out that there are genetic variations in genes called as GATA2 and PCDH15. Again, the names are not important. The concepts are. These GATA2 gene and PCDH15 genes, they have genetic variations, and these variations lead to the different musical ability, the different musical receptivity of two different individuals. If these two genes are expressed, where these are expressed in inner ear, in the cochlea, in the inferior colliculus. So, what do you assume from here? What do you presume from here? It is these particular genes, they code for proteins which change the vibrational energy that is going inside our ears. They change it to certain other form. The proteins with genetic variants cannot change it, or to change it to different forms. And thereby, I may not like Rabindra Sangit because that is being converted to a completely different form because of a variant in this gene. The other person might love Rabindra Sangit because this conversion was not occurring. This fascinating musical ability is with respect to the genes. So a new field has been developed called as biomusicology and evolutionary musicology, where a lot of studies have been going on the biology and evolution of music. So I'm sure that at least some of you are conversant with this particular crow. This crow is a three-eyed raven in Game of Thrones, the Game of Thrones series. So this is supposedly a mythical creature, but not. You can find this for homeotic mutations. Similar cases actually occur in case of Drosophila, where legs maybe form in antenna position. Now this is a snapshot from a famous trilogy called Lord of the Rings, where it was shown that different types of hominoids, like the dwarves, the elves, the humans, the orcs, they all coexisted. Now, what is this to do with genomics? Now we have discovered by studying the prehistoric organisms, by sequencing the prehistoric, like the prehistoric humans, the Homo neanderthalensis, the Neanderthal men, the Homo denisovans, the Denisovan men, Homo floresiensis, the Floresiensis men. We have sequenced all of them. We have compared their sequences with Homo sapiens, and we found that actually all of these Homo sapiens, Homo neanderthalensis, Homo denisovans, Homo floresiensis, there was one time on Earth when all of them were existed together. And to make things more interesting, these Homo floresiensis were short in height. So were they the hobbits? Were they the dwarves? We don't know. Okay, so this is the Neanderthal Genome Project, which is still now going on, where from the fossil records, people are trying to figure out what is the sequence, how big is the difference in the sequence between Homo sapiens and Homo neanderthalensis. One of the most interesting questions that is still still hidden from us is how come Neanderthal men became suddenly obliterated, became extinct? Because prior to that, for 40,000 years, Homo neanderthalensis and Homo sapiens mated between themselves, and a genetic exchange occurred. What happened? So all these are different avenues of genetics and genomics, which is waiting for bright kids like you to take up. Okay, so you must be thinking, what sort of question is this? After so much of blabbering about human genetics, now I'm asking that, so you think you are human? There's a reason behind it. So suppose that this is the total number of humans in Earth today, but this staggering number, which I will not even I do not have the audacity to read out. This staggering number is the number of the microbes, and the microbes are everywhere. They're on this floor, they're on your kitchen sink, they're on your chair, they're on your mobile phone that you're holding now. And yes, they are on you, inside you. Right at this moment, you are harboring 100 trillion bacteria in your body right now. And is it only 100 trillion bacteria? No. You're also holding their genes, their genomes. So you are not human. You are a walking bacterial colony. You are helping the bacteria to grow. That means the bacteria has some importance. We all knew that gut bacteria, they help us in digesting the food. But what if I say that the same gut bacteria help in our mood swings, help in our behavior, even help in protecting us from schizophrenia? So we have used two words. One is gut feeling. We didn't know that why we are saying so. Now we can understand. Oh, is this the etymology? One who framed this gut feeling thing, did you know that actually the gut can feel through the bacteria? And another thing is a common Bengali notation, pet kharap na matha kharap. What is the relation? We know now that if someone does have an upset stomach, it might have a repercussions on your brain. Thereby, pet kharap and matha kharap may happen together. Thus, our microbiome, the gene that is contained within the bacteria is present within us. Our microbiome contains 100 times more genes than we have. They form our second genome. And this paper came out in Nature. You must have heard about this famous prestigious journal where the microbiome genome inside us was given the name our second genome. So it looks like each and everything that we are discussing is determined by genetics. So there is a muscle-related gene which helps a dog to run. So is that gene present in the human? If it is present, if the genetic variation is present, then will he or she be a good athlete? The presence or absence of baldness, high IQ, low risk of Alzheimer's or breast cancer, 20/20 vision, all of these depends upon these blocks of ATGC, the combination of ATGC. So are we prisoners of our genes? We cannot say that, because it also depends upon our lifestyle, our environment, and things like that. So we might have an inherent tendency to be obese, but if we do not eat, then we will not be obese. Similarly, we might have an inherent tendency not to be obese. But then again, if we continue eating binge foods, might be obese. So they say that genetics only loads the gun. It's the lifestyle, it's the environment which ultimately pulls the trigger. Let's get back to this particular individual, Renan Ostuk. I said that why take the risk? Why he took the risk over his, over the concern of his death? Was this risk worth? He might have died. Then if he'd survived, then he could have gone there maybe 5 years later, maybe 10 years later. Why did he take the risk? Because risk taking is cool, as we say. Well, it is. Risk taking is cool evolutionarily. Evolution has guided us, evolution has pushed us to take risks. And this is related to something called as sexual selection. Risk taking is a sexually selectable trait. Not every individual will take the risk. Thus, risk taking is not a trait in everyone. If the risk taking is present in a specific individual, then it will be favored by the opposite sex more than the other individual who does not take a risk. This is not a universal truth. So whoever in the audience is thinking that I do not take risks, so am I not be I'm suitable for boy or a girl for a opposite sex? No. You don't need to think like that. It's not so oversimplified. So sexual selection explains traits that seem to have no survival benefit as such. But fitness thereby isn't just about survival, it's about reproduction. So a person is very fit, but he did not reproduce. The gene got lost. So the ultimate, the benefit of sexual reproduction is the passage of the genes to the next generation. And thus, for reproduction, you need to attract mates. Thus, sexual selection is a very important thing. So the classic example is peacock's tail. Because of this wonderful ornamentation of the tail, the peahen comes, mates, and the next generation happens. And this is true for all the organisms. So what we might not know, that this was the second part of the study that Charles Darwin did. And after his first book on natural selection, he published another book on descent of man and selection in relation to sex, where he said that musical ability is a secondary sexual character in human beings. Because if a person is apt in music, then the chance of attracting the opposite sex is better. Again, I am also musically dwarfed. So those individuals who are not a singer or do not love music, there's nothing to worry about. There are a lot of many other things by which you can attract the opposite sex. So as I said, this fun-loving nature, music, looks, brains, etc., are all sexual selectable traits. And this is also selected. Last but not the least, I would like to say something about nightmare and dreams. We all know that we have this nightmare, we have these dreams, but till date, there was nothing genetics associated with it. Till the last year, when people found out that if you delete the muscarinic acetylcholine receptors called as CHRM1 and CHRM3, you are obliterating these. Then what happens is the non-REM sleep. You must be knowing about REM and non-REM. These are two different phases of sleep, and we dream during the REM sleep. So if you obliterate the CHRM1 and CHRM3, then what happens is the REM sleep part completely gets obliterated. So a mouse is surviving only through non-REM sleep. And thereby, the mice are surviving without dreaming. Because dreaming is a condition that has to occur during the REM sleep. But by deleting the CHRM1 and CHRM3, you have obliterated the REM part of the sleep and thus the dream. Thereby, this is now being touted as, is this the dream gene? So people like me and many others are trying to formulate a project to find out the genetic difference of CHRM1 and CHRM3 in random population and to get to know that what sort of dreams they have. And to assess that, okay, he remembers the dream vividly. He sees colors in the dream. She doesn't see any color in the dream. Can we associate these parameters with the polymorphisms in CHRM1 and CHRM3? So I hope that I have given you enough avenues to explore and to not to confine only in disease genetics. When you say something about, when you like dream of reading genetics and genomics, dream of like studying in your PhD, in your master's, higher studies, genetics. I will end up by showing this slide. Here you can see that a person is standing on one leg, but there is a like somehow or the other there is an impression. There's nothing like that. This particular person has an amputated leg, but then just now he will be having an itch on his right leg's thumb. How is it possible? Or right leg's ankle. How is it possible? He does not have the right leg. This is called as phantom limb. When you are having a lot of sensation on an amputated limb, you do not have that limb, but you are having tremendous pain. So this was first identified during Second World War, I think, when a person who was like a doctor who was like adding morphine to who was giving morphine to a patient whose leg was just then amputated. The patient was screaming and saying, "I'm having terrible pain on my right knee." The right leg was entirely amputated. So this is called as phantom limb. How does it happen? It is you who will think and tell the others. That is the like take-home message. So I will end up by like the famous Ritik Ghotok quote, "Bhabo, bhaba practice karo," and that will help you solve all problems in genetics. Thank you.
Thank you, Dr. Sengupta, for your such a nice presentation. Whenever a good researcher or good scientist become a good teacher, that compilation is something really different. That is being proved in your lecture, and the students have really enriched. I also enjoyed your lecture and listened it entirely. From the side of organizer Bijoy Krishna Girls College, Howrah, we are arranging this student development program or advancement program in association with Clarkson University, USA, as well as Cytogenetics and Genomics Lab, University of Calcutta. You know that. I have a verbal conversation with you, and I am really very happy that you are here and you have enriched their knowledge in such a nice way. And your slides were very attractive, I must say. Now I want to ask you, there are so many questions. Will you like to answer few of them from the student side?
Yeah, sure.
I think more than 15 questions are there. Will you please like to take few from them?
I can take about 5 or 6, but
Okay.
rest I also like to know that what are the questions. So if it is possible to email all the other questions to me, I will be happy to answer each and every question to the students.
Okay, first of all, I would like to request you to stop sharing the screen so that it will be easy for us to handle the questions. Thank you. So I'm taking few questions and then I will mail you. Most of the students have actually appreciated such a nice talk, sir. It is an honor to have you and all this. First question, sir, is there any, sorry. Okay. Is the presence of gut microbes and salivary microbes in human body overcompensate their genome? And in personalized medicine, is this how these microbes plays a vital role?
Oh, it's a fantastic question. I think by overcompensating, I think the questioner wanted to ask that whether there is a crosstalk. There is a tremendous amount of crosstalk, and the microbiome they are known to elicit, they are known to secrete things like dopamine and serotonin. We know that dopamine and serotonin are neurotransmitters that are secreted by our own cells. So you can now well imagine that a person who is having a typical type of microbiome, who are capable of giving rise to a lot of more dopamine and serotonin, it will add on to our own dopamine and serotonin, and thereby our mood swings will be more than a person who does not have those kinds of microbiome. So yes, the microbiome does have an unprecedented sort of a power to alter our behavior and mood, and also mental disorders. In terms of personalized medicines, it is a very new avenue, and I'm glad that you put this up. People now are trying to find out that whether or not the medicines that we take in are also dependent on what kind of microbiome we have. We do not have any ready answer, but this is a very important field that people are looking into.
Okay, the next a little bit relevant question, what I think is, so is there any direct relationship between certain disease and skin pigmentation? Like it said that fair, fat, female, fertile, and unlikely to have cholelithiasis. Cholelithiasis.
So is the question that is there any kind of relationship of the pigmentation with albinism? Is this the question?
No, the question is actually not clear. I've started it, but it is not clear to me even. Because this is fragmented in two parts. Okay, we will send the question. Then another is, let me see. Sir, just like music genes, are there genes that has impact on health of a diseased person?
Of course, and that's the whole thing that like on which we gave a talk, that the genes are there. The genes are the responsible factors of how an individual would be like healthy or not healthy, fragile in terms of their bones, etc., etc. So the entire like medical condition of a human being depends upon the genetic constituency of that particular individual, along with the lifestyle that he or she is like taking.
Okay, seems to be quite labor intensive. Is there any? No. This is not a, okay. Let me see. Sir, can you please explain how to study of gene sequences with modified nucleotide? It will be clear then. I think you can answer this later, because she is actually asking about the procedure, I think.
Okay. So
Okay.
if it was not clear, then you can like get the email from madam and like directly email me your query. I will be happy to send you a video documentation on sequencing.
Then a question is there, which is I don't think very relevant. How do environmental factors affect gene? There are so, I mean, it is a very large question actually.
I mean, that is like.
Sir, you mentioned various diseases are caused by mutations of genes. Is there any mechanism of gene editing so that particular mutation can be reversed? I think Sir has explained this. How does melanin treatment work? I think Dr. Sengupta, you have network problem. I think Dr. Sengupta has network problem. Okay, students, what we can do, we can mail your question to sir further so that he can answer them. Probably sir has network problem. Okay, so right now today's session is over. I think you have your feedback link in the chat box. Is it already given, your feedback link? We are recording all of your questions and we will send them to sir for further answer if possible. Okay. Tomorrow class, the class for tomorrow will start at sharp at 7 p.m. and our overseas collaborator, Dr. Mondal, is going to elaborate the most important statistical technique to you. And I think you will really enjoy them because beside a good scientist, she is a very, very, very good teacher. So I think you will enjoy it. But for today, the meeting is over. Kindly copy the assignment form link from the chat box. It is being provided. Kindly collect it from the chat box. Disconnected.
ম্যাম চ্যাট বক্সে বোধহয় লিঙ্কটা দেওয়া নেই আজকে। হ্যাঁ দেওয়া হয়েছে, দিয়েছে অমৃতা দিল, হ্যাঁ অমৃতা দিল, হ্যাঁ
অমৃতা দি এইমাত্র দিয়েছে।
হ্যাঁ দিল, চার্জ দিল অমৃতা দি,
অমৃতা দি দিয়েছে।
হ্যাঁ দিল, চার্জ দিল, হ্যাঁ।
Okay, so students, we are going to monitor and post few names who are not attending the classes but continuously sending the feedback and assignment. We will declare the names tomorrow. Please, if you are interested, come to the class either in the YouTube live or in the Zoom and then go for assignment. Otherwise, it is not necessary. Okay. So tomorrow class will be from 7 p.m. Even Monday and Tuesday, all those classes will be 7 p.m. If there will be any change, we will post them in the Telegram group. So sharply join by 6:50 p.m. tomorrow. Okay. Thank you. And now you may leave. Okay, you may leave. মইনাককে কিন্তু মানে দেখাচ্ছে রয়েছে, ডিসকানেক্টেড হয়ে গেছে।
পাপিয়াদি থাকবো না, বেরিয়ে যাব?
হ্যাঁ, ইন্দ্রনীল একটু থাক, কিন্তু এখানে অভিনবকে আমি দেখতে পাচ্ছি না।
আচ্ছা আমি ওকে বলছি, আমি ওকে বলছি, আমি বলছি।
আমরা কথাটা একটু বলে নি, আবার নতুন করে ঢুকবো না।
ঠিক আছে, ওকে ওকে।
All participants kindly leave. We have a meeting. অদিতিকেও একটুখানি ফোন করো, আমার ফোনটা কোথায়? So we can stop streaming over here. Okay, বেরিয়ে যেতে বলবো।